Good afternoon, everybody. Today I will be presenting a study based on amplification-based approach for whole genome sequencing of SARS coronavirus 2. The original research article is named as AMPLICOF, Rapid Whole Genome Sequencing using multiplex spatial amplification and real-time Oxford Nanopore sequencing enables rapid variant identification of SARS coronavirus 2. I was not part of this research team, but this research was carried out in Robert Koch Institute at a highly pathogenic viruses division under Professor Andreas Nietzsche's supervision. So as you know, there were two SARS coronavirus cases. The first reported cases was in 2002, November, the first SARS coronavirus case. It took almost 6 months for at that time to give the whole genome sequences, and it was like 30th May 2003 when they could finally sequence the whole genome sequence of the SARS coronavirus. But in the recent outbreak in SARS coronavirus 2, the first case reported in 31st December 2019. So with the advancement of the sequencing techniques, we could the scientists could do the whole genome sequencing almost within a week. So they could release the whole genome sequence of the virus by 9th January 2020. So why do we need the whole genome sequence? With the whole genome sequence data, we could do the phylogenetic analysis, and we could also do the transmission train tracking, and we can identify possible therapeutic targets. We can do a timely risk assessment, and also we can identify novel variants. So basically, there are two sequencing platforms that are widely used nowadays: Illumina sequencing platform and Oxford Nanopore sequencing techniques. If we quickly look into the advantages and the disadvantages of these two techniques, in Illumina technique, you can get very high quality reads, and also the accuracy rate is higher than the Mina technique. The disadvantage of the Illumina sequencing techniques is the equipment, reagent, and consumable cost is very high, and also the cost for the data storage. And when we're using the shotgun approach, we also have some issues with personal data protection. So in contrast to that, if we take the mean-ion sequencing method, the advantage is that it's a portable device, and we can do the sequencing in real time. You can see the reads in number of reads you get in real time, and the equipment cost is low, and also the reagent and consumable cost is low compared to the Illumina sequencing. But the disadvantage is the read quality is a little bit lower than compared to Illumina. So why do we use an amplification-based approach? So if when we are amplifying, we can get a high read coverage for the genome, and also we can identify the SNPs, insertions, and deletions. And of course, the specific amplification of the virus genome can exclude the data protection problems we get from other sequencing methods. If I quickly go to the overview of this research, they have used two reaction multiplex PCRs, two primer pools with 113 and 111 primers, and they have established the AMPLICOF PCR, AMPLICOF assay using SARS coronavirus 2 propagated cell cultures, and also samples obtained for the external quality assurance exercise. They call it INSTANT. So they did the establishment with those samples, and then they analyzed the clinical specimens to prove the applicability of this assay in routine diagnostic settings. Moving to the materials and methods. So they first designed the primers using primer 3 application, and they have use the all reference sequences available at GSED, and also NCBI, and they aligned the reference sequences, and then primers were designed on the conserved region between the SARS coronavirus 2 and the SARS coronavirus to enable the amplification that they assume that possibly arising mutations in the future. So as I said, the primers were designed to cover the whole genome, And the primers were separated into two pools, 113 and 111 primers. So they have different amplicon lengths, 119 and 775 base pair lengths. And they have a optimal melting temperature. The primers have optimal melting temperature around 60 degrees, minimum 59 and maximum 61. And the resulting amplicon length was 250 to 650 base pairs. So if I give you a quickly explain what they did. So they have they extracted the viral RNA using collagen viral RNA minikit, and then you do the cDNA synthesis. They have followed the superscript protocol for cDNA synthesis, and then moving on to the PCR amplification. So since you have two primer pools, you prepare two master mixes, and then finally you, after the PCR, you pull the primer mixes together. So the amplicons, you pull it together. So for the evaluation of the amplicon sequencing, they have three different settings. So the first one was the cell culture propagated virus strains. They have used three viral strains from Italy, Germany, and Japan. And they have used 36 clinical samples from patients with clinical and laboratory diagnosis of COVID-19. And then they have the third settings with the quality assurance samples for the quality assurance from instant. They had the inactivated samples for sensitivity testing with different concentrations and for specific assessment with other coronaviruses. So they have compared the two sequencing platform, the Illumina sequencing platform and the Oxford Illumina sequencing platform. The library prep for Illumina, they used the Paragon Twinplex assay and did the sequencing on MySeq. And for Oxford Illumina technique, they have prepared the library using two kits, a rapid barcoding kit and ligation sequencing kit, and then they did a comparison between the kits, and they did the sequencing using spot on flow cell. So as I mentioned, they had three different settings. The first one is the cell culture propagated viral strain, three strains from Italy, Germany, and Japan. So for that, they use the rapid barcoding kit. And then they have the clinical samples, 26 samples who were suspected as COVID-19 patients. They use the rapid barcoding kit and the ligation sequencing kit. And for the quality assurance purpose, they had different settings. So they use the ligation sequencing kit. So they did the bioinformatic analysis. You get the raw data as FAST5 files, and then it was converted to FASTQ files using the MinID device. And then you can also choose to demultiplex the reads, and also you can trim the barcode, also using the Mino software. And if you need further trimming, you can use software like POCHOP. And then the trimmed FASTQ reads will be aligned to, it was aligned to reference sequence, and the consensus sequence was called using Medaka. For each sample, they generated 50,000 reads, and all the sequence data you can find at the European Nucleotide Archive. So as I mentioned, they had three different settings. So these are the results for the three different settings. They had 75,000 reads for the cell culture propagated virus strains from Italy, Germany, and Japan. It had more than 98% of the genome coverage, and the depth was also more than 10, and it was identical for the reference genome. For the clinical samples, they had 50,000 reads per sample, and the genome coverage was 92 to 99%. The depth was more than 10. And for the instant samples, the genome coverage was between 94 to 99%, depending on the CT value. Then they tested the specificity of the assay. They had different samples tested, HKU1, NL63, OC43, and 229E. There was no amplification there was no amplification showed. Those are also different coronaviruses, but there was no amplification shown. So it confirmed that this assay is specific for the SARS coronavirus. But it showed amplification with low coverage with SARS coronavirus. So this means that you can use this assay even with the increasing mutations with the SARS coronavirus too. So all amplicons of the SARS coronavirus still could be distinguished from SARS coronavirus 2. That means it eliminated the false identification. Moving on to the sensitivity of the assay. So to test the sensitivity, they tested it with different virus loads, but samples which had a low viral load, which means if the CT value is more than 31, those samples could not be sequenced with the rapid sequencing kit. But if you use the ligation sequencing kit, you could sequence samples with CT up to 35. But as you can imagine, if you have low viral load, it takes a lot of time to generate 50,000 reads. So the sequencing time increased from 1 hour and 30 minutes to 16 hours for samples with CT 35. So if I compare the two library prep kit they used in the study, they used rapid sequencing kit and the ligation sequencing kit. So as I mentioned, if the virus load is low, the rapid sequencing kit is not suitable because it takes longer time and not working with the CT more than 31. But you get a genome percentage coverage slightly higher than ligation sequencing kit. So it's the opposite with the ligation sequencing kit. You can go up to CT35, sequence time was a little bit less, and the coverage is slightly lower than the rapid sequencing kit. So this is the table from their research publication. As this is actually what I summarized. You can see the percentage of the genome coverage for each sequencing kit. This whole thing is for the rapid library kit. This is with the ligation library kit. So these are the CT values. The first sample had a CT of 18 and the genome coverage is 98.4, while the ligation kit had 97.3. So as the CT value increases, you can see for the CT24, the coverage is 94, and ligation, its coverage decreases to 91%. And this is also another table showing the clinical specimens, what they received, results received for the clinical specimens when they use, this is when they use the rapid library, and this is when we use the ligation library. So because those are clinical samples, you cannot actually predict what type of CT value you get. So you have to pool sometimes samples with different viral loads. So some patients might have low viral loads, and some patients might have high viral load. That means very low CT value. So yeah, when you're pulling together, you cannot predict what like you have to pull it together in clinical specimens. These are the samples they received for the external quality assurance purpose from INSTED. They as you can see, they have the genome coverage from 94 to 99%. And also they were able to identify the sequences accurately using the AmpliCuff assay. This is again the 3 different strains received from Italy, Germany, and Japan. You can see that the number of reads increase over time up to 75,000 reads within 1.5 hours. Other than these samples, they also did use the AmpliCuff sequencing assay for 3 other patient samples with CT values 21, 21.4, 20.9, and 32.3, which are suspected to be the UK variant B117. And they wanted to see if the assay is working, and they for that variant, and they could detect, or they were able to identify 8 out of 9 mutation occurred in the S gene in variant B117. You can see it from the next slide in the figure. Here you can see the three samples, sequence 1, 2, and 3 are the three patient samples who were infected with the B117 variant. You can see that the mutations in sequence 3, you can see all the 8 mutations except for this B118H, which was not detected by the Amplicov assay. So apart from all this, they also wanted to test if we can use this to explain virus. If we can use this assay to explain the viral transmissions. So there was an incident where there were a positive patient in co-working space. So they did a PCR screening for all the workers in that settings and found out there were 2 additional people who were positive for SARS coronavirus 2. And then they did the genome sequencing, whole genome sequencing for these 3 patient samples. And then they found out that there was no relationship between the 3 cases pointing towards independent infection event. And they also did the same thing using the Illumina sequencing method, and it confirmed the Amplicov results was accurate, was correct with the method inherent delay. So that means we can also use this to explain the virus transmission. Moving on to the discussion. As you know, the whole genome sequencing has become an integral part of outbreak investigations. Genomes are being added daily and have been made available in GSET. Its genomic surveillance relevant for outbreak investigation, including virus diversity, their virulence, and the transmission route, possible mutation, identification of hotspot transmission, improvement of molecular diagnostic tools, and therapeutic targets. If we check the relevance of the whole genome sequencing during viral outbreaks, this was not the first time that they used the whole genome sequencing used in a viral outbreak. They have used it in Ebola outbreak in West Africa in 2013 to find that the mutation arised because of the host adaptations, and also for the Zika outbreak happened in 2016. It showed that it had multiple introduction events in the Zika outbreak when we do the whole genome sequencing. And for the current SARS coronavirus outbreak, it enabled the calculation and analysis of the phylogenetic trees and the receptor binding surveillance. And you can use this for updating the in silico binding of the diagnostic assays. For example, like if you want, the current diagnostic assays were introduced at the beginning of the outbreak. We are still using it, but if there's a new variant, we can do the whole genome sequencing and see if our diagnostic assays are still working to detect the coronavirus patients, COVID-19 patients. And also we can use the 3D protein structure modeling and also to investigate potential drug targets. And very important to identify mutations. So advantages of using amplification-based method, I explained this as I explained earlier. Sequencing of the viral genome directly from patient samples, it can give you a huge background noise, a high amount of human background sequence. You get high amount of human background sequences, and also to achieve a full and deep coverage of the viral genome, it takes a lot of time, and it's also cost intensive to generate millions of reads. So so these are the advantage of using amplification-based method. So in this study, they have reported multiplex PCR and MinINE sequencing approach for whole genome sequencing of SARS coronavirus 2. It can provide sensitivity up to CD35 whole genome sequencing when we are using ligation sequencing method. And sequencing time takes only 30 minutes until almost complete genome coverage. And whole genome sequencing of SARS coronavirus 2 directly from patient material eliminates time-consuming culturing steps. So you can, it's very efficient and also accurate. So this approach offers you, in comparison to other commercially available assays, this Amplicoff approach offers a flexible usage of primer pools composition. You can, since you design the primers and you know the primers where they bind and everything, you can support the primers targeting only the S gene for specific questions. And also it reduces the PCR time. The primer anneals uh, 30 seconds and annealing time for primers instead of 5 minutes. And also it reduces hands-on time using this assay. So in conclusion, genomic sequencing is an irrepressible tool for real-time outbreak investigation, including the initial pathogen detection and characterization, then transmission train tracking, protein modeling, identification of specific mutants, and also Amplicoff eliminates time-consuming culturing of the material, and it also ensures the personal data protection, and it is very affordable and field deployable technique. It can be used by laboratories with limited budget in remote areas. That's why this is a very good method to use in our labs in developing countries. So I would like to thank Robert Koch Institute Germany for, as Dr. Sampath mentioned, we are using this assay in our lab to sequence the samples from Sri Lanka. So the necessary training, all the equipment and the consumables for the initial establishment of these assays in Sri Lanka, everything was provided by Robert Koch Institute. So I would like to convey my sincere gratitude, especially to Professor Andreas Miche, the head of the highly pathogenic virus division at Robert Koch Institute, and Dr. Claudia Call, Dr. Annika Brinkman, and Mr. Stephen Uddin for their kind support. And also all the IDEA project members in Sri Lanka and in Germany, as Sampath mentioned. And also, thank you so much for the invitation, Sampath, and thank you for coordinating such an event.
Yeah, thanks, Thejanee. So yeah, the floor is open. Thejanee, you can lead the discussion. Yes, I mean, what you can do, you can either type in the chat box or you can directly ask questions.
Thejanee, there's a question already in the chat box.
Is it possible to increase the read quality in MinION by using a good workstation, as in good spec computer?
Yeah, the read quality does not depend on the computer. It's you get the MinION device. Actually, we received the device in 2019. They had the MinIT device, which are compatible with the MinION device. Now you can get MK1C. It's inbuilt. You don't need a separate computer for this. So everything happens inside the MinIT. So they incorporated these two devices together. Now you get MK1C. It's one equipment, but it does not depend on the computer read quality. It depends on the library preparation steps or the kits you are using, and also depending on the quality of the samples.
Thejanee, a follow-up question from Bhushan. It says, will using a IC during RNA extraction be a problem for sequencing? So he's asking that the using of IC during RNA extraction be a problem.
No, actually, as I said, sir, in this method, they have used the primer specifically designed for the SARS coronavirus. So we are amplifying only the SARS coronavirus. We are not amplifying anything else. So no, it's not a problem.
I have a question, Thejanee, in relation to amplification. So, so the primers. So you. For this particular case, for SARS-CoV-2, the primers are designed separately. So if you were to use so wild type, not a virus, but something like a say another organism. So are there a general set of primers that normally we use for Illumina would work here, or are there specific primers to be designed to fit into Min9 system?
You can design your own primers. So I think it's better, like, for example, if you're doing a species identification, you can use.
No. For a like a broader genome wide, say, like we use APK, we use PST1, like, standard general purpose primers that works well for Illumina. I wonder whether would the same primers work here. These are like basically you just chop the genome by going all over the place. So would that work here?
It should work, sir, because it's the, as you said, it's the primer should work, but you have, like, you can use the only difference is you use a different library preparation kit, and so you can ligate the barcodes for each amplicon, so you can use the same primer.
So the library prep step will basically prepare the sample. So when you do that accordingly, the Min9 will take it, right?
Yes,
yes. That's interesting. Thanks.
Thank you, Thejanee, for the very nice presentation. So basically, I have a few questions regarding the library preparation. Now, so with this mini-ion kit, so that means, do they provide the already updated libraries or any kind of existing genomes? Or is that, can we do the customization in that library preparation step? That means, like, we can find most updated, recently published. Yes.
I think what you
recently
want to
published,
ask is,
can
like,
we add, for example, we
are not changing the library kits. We can change the primers. That's what they have also mentioned in their essay, that when you have your own primer pool, you can change the primers accordingly. And then, for example, if there's any variants, and if you in silico, if you see that your primers are not binding with the specific region, you can change your primers, not change, but actually you can add more primers to your primer pool. Is that what you asked?
Yeah, because with the over the time, so the virus getting evolved, and like maybe you need much more, we have to increase the libraries, right? Because the more and more variants are coming.
Also
maybe
then I need to add something too. So that's why they also, like, in the first place, you actually choose more conserved regions to design your primers. So,
okay, so and thank you very much for that question, that answer. And
so
the other one is, like,
if you still see that there are nuances,
I got the point. Yes. And the second one is, can we use some marker genes only because rather than going into that whole genome sequencing,
like, you mean the S gene
homologous?
Yes, you can do the sequencing of S gene, because you see all the mutations there, but it's advisable that you first get the whole genome sequence, and then you can go for you can see the mutations specifically in the region.
So when we do this workflow, or the bioinformatic workflow of this Min9, so that means, is that possible to take the raw data from which is being sequenced at the first stage, and do it by our own platform, maybe using some, like, there are other bioinformatic. I just wanted to know about that, rather than going with I know that they are providing
Yes, that's like, you can find like something like a PowerShop in GitHub. You can find other bioinformatics workflows. It works with any bioinformatics workflow if you have the raw data or the FAST5 files.
Okay, thank you very much. Thank you very much, Thejanee. Okay.
You're welcome.
Thejanee, a quick question regarding the data storage. You mentioned earlier that compared to Illumina and other sequencing platforms, NanoPro is a bit safer because of the storage related issues. So is there a cloud provided by NanoPro, or I didn't exactly understand how this data is kind of better stored in your system. So how, when you generate the reads, where you normally store? Is it in your
It's
own
usually by
server?
default, like it's stored in this MinIT device.
I see.
Yeah, it's
So it has a storage capacity
yes,
of,
it does.
of how many terabytes?
I don't remember the exact number. So it's have a.
But it comes. I see. All right.
Yeah.
Go ahead. Yeah.
It's not a cloud-based, so it's.
I see. So that's actually helpful, because when it is a cloud, then sometimes data accessibility and the internet related matters also come into play, right? Yeah. So no, I believe you. I just wasn't sure how it works. Yeah.
I think on data security, what Thejanee meant was you're not like you're getting specific sequences, not going into the whole sample, right? In that case, you're not actually saving all the human data and all that also.
And Sahan is saying that it's 1 TB.
It's 1 TB.
And 512 GB. Yeah, and I think the beauty here is that you don't get.
So you need to offload.
Yeah, you have to offload, and then in the viral in the COVID SARS-CoV scenario, the genome is very small. In a mammalian or sometimes in amphibian genomes they are huge.
Yeah.
But then you can always offload it, right? Yeah.
Yeah.
So, and also I think since you get a very real-time sequencing, you can get what you want and terminate the sequencer.
Yes.
Right?
Yes. That's also
in Illumina. The problem is you get about 300 GB in one run, and then you don't know what to do with it. Yeah.
Thejanee, thank you for that elaboration. It was very useful. What is the status of the work that is going on with Professor Inoka these days?
Madam, we sequenced 28 samples the day before. We actually received already extracted samples from MRI, and then we proceed from the cDNA synthesis, and then we do the PCR, and then we sequence the samples.
Right. So are these blasted against the other sequencing method? Do you have sequences?
Yes, madam. We blast against the reference sequence. Yes, madam.
So now we are ready to, so Professor Inoka, then we are ready to tell everybody that you have this sequencing recipe. That's what I was trying to get at.
It's safe to say, madam, yes.
Right. Okay, that's great.
Yeah. We've been doing sequencing since last year, but now only it has taken up into a more kind of routine way.
Right, right. So these variants that are coming up, what do you have to say about this? What the locally we are generating new variants, right?
Madam, is this for me? The
question? Yeah, anyway, yeah, maybe. Yeah.
Okay. Yes, we didn't find any epsilon, so it's only at the moment it's only the Delta variant and the UK variant is circulating here.
Okay, right. Okay.
I think regarding variants, the issue is now if you go through the genomes, you will find enough variants, but whether they are clinically significant is the question.
Right, right. So it's just for transmissibility, is it? That the variants are important. That's what I understood.
Pathogenicity also, madam.
Right. Okay. Right. Transmissibility and what do you call it? Virulence.
Virulence.
Right. Thanks for elaborating. Thank you. This is a good eye opener. Thank you so much, Thejanee. All the best to your team for this effort. Right.
Thejanee, in relation to variants may so that so yes, obviously there are so many non significant variants. And so when you find a variant, so what you do, you run the protein code and to see whether there could be functional changes, and would you be looking into specific regions like in the spike protein and all to see whether there can be potential changes, alterations in its virulence and so on? Are there any kind of formal methods there to detect these variants, or you'll wait for somebody else to do that if you detect variation?
Yeah, you can use a software called Pangolin, sir. It gives you the variant already. It's like when you upload it, it blasts it against the reference sequence they already have in the databases. So it already gives you the results which variant you have. Like if it's also you can manually by looking at the, as you said, we can look at the gene sequence, and also from this study, if you remember, sir, they did sequence for 3 samples for the UK variant, and there are the mutations, they could identify 8 out of 9 mutations. So you can see the mutations in the sequence manually, or you can use a software like Pangolin.
There's a question, Thejanee.
Yeah.
Rajita Anuruddha has a question. Do you have any data accuracy comparison between Illumina and NanoPro?
They didn't specify any number for the data accuracy percentage in their publication, but if you want to know more details, I can ask the research team and let him know.
And you can run multiple species simultaneously, Thejanee, because you have the barcoding, correct?
Yes, multiple samples we can run multiple samples at once. We usually run multiple samples at once.
But at this stage, you cannot, I guess, because the equipment is for some other purpose, is it? Like, for example, if I wanted to sequence a section of a gene from another organism, is there a kind of processing like procedure in place that I can pay for that and get it done, or would, like, this is a real question. Like, I wanted to sequence sections of DRD4 to see, the variations, the polymorphism. Is there a procedure in place to do that, or do I have to wait till another Minna and NanoPro showed up, from your lab or from somebody else's lab?
No, actually we can. Yeah, we can use already, like we usually use a flow cell 4 times after pooling the samples. So because the number of pores in a flow cell decreases over time. When you run a lot of samples, the number of pores available for sequencing reduces. So we usually don't use the flow cells after 4 runs, but if you want to do a testing run, you can use a already used flow cell. It wouldn't affect your work because it's the coronavirus samples, so it wouldn't affect
that's what I
so
was
you can use that flow cell here.
So you will have the 5th and 6th runs like free of charge, is it? for people like us?
Yeah.
All right, good to know. Sahan is saying that you can use Pango lineage online platform or NextStrain online platform for variant identification. Thanks, Sahan. So right now in the lab, so you, Sahan, and Inoka's team is leading the analysis. Are there will there be a formal technician or somebody get trained for this work for the long run, Inoka, or Thejanee can answer just for kind of technical clarification?
So for the continuity of the lab, we actually requested for 2 dedicated RAs
Nice.
Because this work now, even every run, we would have something that we need to kind of, technically look at with. So we wouldn't think a technician would be able to actually handle all that. I mean, for the continuity, we thought of actually, even now, Sahan and Thejanee, they can't be doing this because they have their own work.
Yeah, their own research work neither to move forward.
So that's why we thought of getting two dedicated RAs trained for this purpose. And then, so we actually get a feedback from the from RKI also on the sequencing. So we do cross-checking, and because it, I mean, the suits that you use, the softwares that you use in order to analyze these, so you need to cross-check these also while you're doing this.
And actually, sir, that this became to spotlight with the outbreak, but actually RKI offered me training in 2019 for this V9 sequencing. So it took a bit of time, like to get this, to get used to handling the equipment and doing the experiments. But yes, so it will take a bit of time to train another person.
Yeah, that's the thing. I mean, people complain about not having the vaccine in time, not having knowledge in time. But the thing is, as a country, if you don't invest ahead of problems, if you sit down and cheering for something else, and then when you get this, then you don't have that much time. So in your case, also you were, they are way before this pandemic, right? Doing that, so that vision has to be there, to benefit from science, right? Otherwise, we have to pay huge amounts and get whatever the scrap, leftover, whatever the science from other places. So yeah, exactly. Very exciting stuff.
Thejanee, thank you so much for accepting our invitation and sharing your experience with us. And thank you very much for everyone for joining with us today. And stay safe.